Hello everyone, and welcome to this audio podcast. This audio podcast today, as part of the series The Quail N Crux. This audio podcast is part of the Ricochet and Ruckus. This audio podcast is part of the Bedlam and Bostics. This audio podcast is part of the Shitstorm Squadron, and this audio podcast is part of the ZtormLabostix Production. Today, I'm looking at as part of this audio podcast. Excuse me, as part of this audio podcast series. A look at the molecular basis of Huntington's. So Huntington's is evidence as an inherited neurodegenerative disorder or condition caused by a single mutation in the Huntington's gene, located primarily on chromosome 4. On chromosome 4, and the wild type Huntington's gene contains in around 67 exons and produces at least two main messenger RNA or mRNA transcripts of different lengths due to the variations in the three untranslated region. Protein functioning in around the Huntington's sort of molecular on the molecular basis function versatility have function versatility which is further sort of modulated by extensive post translational modifications such as phosphorylation and ubiquitination and sorylation, which are influenced by protein expressed on location in around the Huntington's chromosome, and where there is protein to protein interaction, and there is then where there is influence in on the the sort of molecular structure of Huntington, on the molecular structure of the Huntington's gene, where there is where the gene then encodes the large Huntington protein of approximately encodes on protein of approximately 3,144 amino acids with a molecular sort of weight of about 350 kilodaltons, and typically containing around 23 sort of linkages of amino acids, cytosine, adenine, and guanine, and then additionally where there are trinucleotide repeats in the on the coding region of the exon. Huntington's is also caused by mutation to the expression of the amino acid cytosine, adenine, and guanine repeats beyond the normal range. The amino acid repeats and length increases over time. The expansion of the amino acid repeats in the amongst the Huntington's disease protein generates a long sort of polyglutamine tract in on the protein N terminus. Alleles with around 26 or fewer repeats are considered normal in the amongst the Huntington's gene, whilst 27 to 35 repeats are intermediate, and 36 to 39 repeats on the amino acids may result in Huntington's in some individuals, whilst others do not. And then 40 or more repeats in amongst the amino acids, those particular amino acids lead to the development of Huntington's disease specifically. From non-effective recipients who have cytosine, adenosine, and guanine repeat towards the high end in the normal amongst the normal range, have shown affected have been shown to have been shown and have been where evidence informs that those infected also are in amongst children or young people in around 10 to 15% of Huntington's cases. However, some studies or investigations have also shown late onset of Huntington's, even with lower number of cytosine, adenosine, and guanine amino acid repeats, such as that between 29 to 34 of and in around the particular amino acids. The number that draws this audio podcast to a close. This audio podcast as part of the Quail N Crux. This audio podcast as part of the Ricochet and Ruckus. This audio podcast as part of the Bedlam and Labostix. This audio podcast as part of the Shitstorm Squadron. And this audio podcast as part of a ZtormLabostix production.